Okay.
All right.
Thank you, sir. A very good afternoon to all. I am Anisha Shingupta and I am very much pleased to be the moderator for this session also, the 5 days FDP program on the art and craft of scientific and technical writing from A to almost Z. Our school, the School of Natural, Applied and Social Science of MAKAUT WB, with the Language Lab and the Center of Linguistics, MAKAUT West Bengal, are jointly organizing this FDP in collaboration with Islamic University of Science and Technology, Jammu and Kashmir, sponsored by Ekip 3. Due to shortage of time, I am going to introduction directly. The session titled Everything You Always Wanted to Know About Impressive Graphs, Figures, and Tables, but were hesitant to ask, will be chaired by Dr. Shoham Mukherjee. Dr. Mukherjee is a postdoctoral research scientist at the Uppsala University at Sweden. He did his PhD from ISC India. He has completed a couple of PDF in several other institutions and has about 17 research publications in international journals. We all have heard the saying that a picture speaks 1,000 words. Today's top-tier journals adopt a newer format focused on broadening the scope of readership of an article. The current format offers the authors more flexibility in the usage of scientific language, storyline, rearrangement, and illustration schemes, rendering readability more interesting and intuitive. This session is dedicated to discuss creative ways to communicate one's scientific findings in the most effective manner. Now I humbly request Dr. Mukherjee to please take over. Sir, please.
Yeah. Thank you, Ms. Anisha Gupta, for this wonderful introduction.
Sorry, sir. Sorry to interrupt you. Because we are confusing between two Mukherjees here. We have with us the HOD of our Forensic Science Department, Professor Dr. Arup Mukherjee, sir.
Yeah,
and you are also Dr. Shoham Mukherjee, sir. So that's why the
junior one.
So, sir, before you start, we'd like to listen some words from Professor Dr. Arup Mukherjee, sir.
Thank you. Yeah, go ahead, please.
Great going the MAKAUT team.
Yes, please.
Yes. Dr. Mukherjee, somehow Mukherjees are involved in academics for a long time, traditional time. So it come perhaps automatic to us. And one of my very interesting experience, just to share for the participants, that I started my work like you in the southern India. I mean, visiting places very close to you, ISC and all that. So people in 1980s, when I was going into my research in 1981 or something like that, they were asking, what, Bengalis are all Mukherjee, Banerjee, Chatterjee, any other? so that is obvious, you know. But nowadays, it is.
And sir, did you reply, Hanji?
রেগে যাবেন না স্যার honestly এটা কিন্তু ঘটনা so now ওই সময়ের কথা বলছি but time has changed and now so their confusion is obvious so I am Professor Arup Mukherjee, walked around, and I am actually fascinated by Dr. Soham Mukherjee's area and Dr. Professor Samazdhar's selection of it. There are speaker is already introduced, and I honestly appreciate the topic. Again, sharing some line. It is very difficult for us, and we are not taught perhaps in India, except in prime institutions, how a graph or a representation can communicate so much. How the data actually and truly can be presented not only in mathematical form, but also depicting in pictorial forms, different forms of graphs, I mean, different forms. And even so, seeing a graph just like an engineer seeing a drawing, can visualize a big thing like a big, say, bridge or all that, just seeing a drawing in different platforms, scientific language. So graphs and tables and figures are actually scientific language. We need to know, a researcher need to know a lot on that. So I appreciate on selection of the topic, and given the experience of Dr. Mukherjee, he is a very fit candidate to deliberate on that. And we just present it because the basics are not taught here in most part of India. So it's a very valuable addition as a deliberation. I invite researchers to gain a lot, raise questions, and know more. I'll be learning more too, and maybe interacting. So welcome, Dr. Mukherjee. Please deliberate your topic, and I'll be keenly seeing it also through. Dr. Mukherjee, please.
Yes, that puts a lot of pressure on me, but jokes apart, I would say that this has been a fantastic opportunity, and thank you very much. Like I'd like to start by thanking specifically Professor Sukhendra Samajdar for allowing me to participate in this prestigious event. And like if I had a call with Professor Samajdar, and then we discussed about the importance of scientific writing and technical writing, which in my first email, in fact, I responded to him saying that I think this is a phenomenal topic and we should focus on it more and more, how scientific language can be developed among young researchers, like for all of us who have actually struggled and learned in our own ways, and now we should, it's time for us to share those knowledges to young people so that they can continue it forward. So on that note, I would like to start by thanking Professor Samajdar and Dr. Arup Mukherjee for this fantastic introduction as well. That MAKAUT is organizing such a valuable event, the art and craft of scientific and technical writing. Thank you all the organizers as well, the School of Natural, Applied and Social Sciences and the Language Laboratory, who have helped to organize this wonderful event, and also and of course the funding agency TUKIP, the third phase. So on this note, I'll just start. And do I start by screen sharing or?
Yeah, please go ahead. Please go ahead.
Yes, sir.
Okay. So please let me know if you can see the screen.
Yes, sir.
Yes, sir, it is.
Yes, sir, it is visible.
Okay.
It's fine.
And can you see it full screen now, or?
Now, yes.
Yes, sir.
Okay, perfect. So everything on the technical sides works perfectly fine now. So I start by the wonderful topic that Professor Samajdar has given me. And the topic, as you can see, is everything you always wanted to know about impressive graphs, figures, and tables, but were hesitant to ask. As Professor Arup Mukherjee has rightly pointed out, that we see graphs all around us every day. We are living in a world of data, communication, and then if you do not communicate it properly, you miss out on a lot of things that's going on around you. So I would go roughly to the session outline. It's a 2-hour session, as you already know. I will start by about the scientific writing today, where it stands, the general format of the paper. But there are other experts in this panel who would shed light on this or have already shed light on this, and I'm sure this event will cover all aspects of it. And then I come directly to the presentation, but that's just a brief introduction, very, very short. just a recap and for the other talks to follow. And then I will pick up on some key factors that you should focus on while presenting your data. And there are two things, scientific accuracy and aesthetics. Both are important. With time, a lot of focus has been put on aesthetics to focus on broader readership for all top-tier journals, as I have laid it out in the abstract. But apart from that, I'd also go into some of the details that are easy to miss out when we are presenting our data. So although the talk says everything about impressive graphs, I'll also go slightly out of the line in some areas so that you also understand and appreciate the importance of the reason why it needs to be communicated properly and with accuracy. Always we have to remember that science comes first and the aesthetic comes second. So I have also paid some attention on the scientific accuracy, and more importantly, to make this session a bit interactive and engaging the audience, there are real life examples around us, and then there are quizzes at different sections. So I'll take a break. As you can see, like after I discuss a little bit about resolution, caption, axis, and scale. Can you see my cursor?
Yes, sir.
Okay.
It's visible.
Yeah. So there are quiz break and questions, and each quiz has 5 questions or 6 questions, and I'll try to communicate those questions to the audience, and it's up to the audience what they think. So I'll give a lot of case studies, because I think the best way to communicate about how to communicate is to communicate through case studies. So you look at examples and you see what is wrong in that, what is right in that, and then at the end of the talk, the goal is that you decide for a given set of data, you would know exactly how to present it, and then you're kind of on your own and practice independently. So that is what I would like to achieve from this talk and also from this prestigious event. So quickly, I would go to the first part of my talk, which is writing science today. And I'm sure you're already acquainted with this, but I'll just briefly summarize like what all the kind of things that goes through when you write a paper, so to speak. And these are like just one-liners, so bear with me just for a moment. So the typical format of a paper has a title, an abstract, an introduction, an experimental section, then you have the results, discussions. In a lot of papers, result and discussions are clubbed together in one format. Then you conclude what your findings are, then you acknowledge, give the proper references, and then you provide additional information, the supporting information. This has been the general format of the paper writing these days, and there are changes in each of this format as we have progressed, particularly over the last decade. Significant changes have happened in scientific writing. So, for example, I'll now go through one by one, and I'll just give one-liners, take-home messages, what things should be. Title of a paper should be very simple, attractive, and it should accurately tell what you have done, just the main findings. You should try to avoid flashy words. Just try to keep it simple, and the depiction needs to be very, very accurate. For the abstract, for the abstract, it needs to spark some curiosity why one should read the paper. So it has to be effective and it has to be very short. But then if there are some details that are very, very important and central to the work, it also needs to be presented. So basically, abstract should be, we have all done précis writing at some point of our times in our curriculum in school. So it's sort of impressive, but it should also generate a lot of curiosity among the readers. The introduction, the first thing is it should motivate why the work is important, and then you follow up with what has already been done, what people know, where the knowledge on this particular area stands, and then you add what is new that you are trying to add. So basically, introduction, it should be a brief storyline of the history of the work. If it's a new work, then not too much has been known, but then you can always motivate, like emphasize on the motivation part a bit more to show that why this would be a promising area in the upcoming years. For the experimental section, this has to be very, very detailed. Often people just write experimental sections without too much details because they have done a lot of work and it's hard to put in. You can put in some of that in supporting information, of course, but you cannot miss out significant details. And one very important thing that I find is that there are some details that needs to be mentioned. There are some very smart tricks that people do while making a material or getting the right data. One needs to mention that explicitly in the experimental section, so that the idea is that if someone else across the world tries to repeat those, that person, he or she, should be able to reproduce it. That's the whole point of the experimental section, that you communicate what exactly you have done to the world. Then I come to the results. This, I'm sure the most of the discussions go into developing how to write the result section. It should be detailed, accurate, and chronological. There should be a storyline that the reader should be able to build on himself or herself as he or she is going along. I have separated out the results and discussions just to highlight the difference between the two. But like I said, then what I'm, yes. So, but then in most papers, these two are clubbed together, but I have separated them out just for the sake of understanding the differences behind the meanings of these two. So for the discussion, you talk about the storyline that you are getting directly from the results. So in the discussion also, you reflect back in the lines of introduction. You say what is old and connect your findings to the old, and then move along to generate the scope of your present results that you have shown. So results are basically only presenting your data, so to speak, and discussion is the story behind it. Finally, you have the conclusion where you mention your major findings and then the importance of your work. So there is often a miscommunication. We as younger researchers, like when we were young, we always equated conclusion with abstract, but it is absolutely not the case. The purpose of an abstract and conclusion are very, very different. In the conclusion, you report your major findings and then emphasize on also the scope and the importance of your work. That you do not do too much on the abstract. The abstract is more like it should help you to just have a flavor of what it has done. It should be short and crisp. But the main point is that it should make you curious. But the conclusions can be a slightly longer format. You can even add figures in conclusion. But it is to highlight the importance more. Acknowledgements, you should acknowledge everybody who has helped you on the work, apart from the co-authors, of course, but then also the funding agency. The references should be accurate and relevant. There's an increasingly large number of references which are inaccurate because people do not check the references. So when you're adding a reference in one of a paper, please just go and Google it up and see if that paper exists. Often we have seen them, and then there are reports across the world, and there are TED Talks about this, and you can go and check that there are a huge number of misreported references which doesn't really exist. Then finally, you have supporting information, where you detail out the more detailed aspects of your methods, analysis, additional data. For example, in the experimental section, you have missed out on the details of your chemicals you have used to synthesize the material, but you've just described the method by which you prepared a new sample. But you can prepare the starting, you can mention the starting samples and supporting information, things like this, detailed of your fitting the data that can go in the supporting information if you want. So it's more elaborate, but remember, there's an addendum to what you have already shown. In principle, people should be able to follow the entire format of the paper just from the experimental section and the results and discussions. Supporting information, as the name suggests, should really be supporting and not the main. So I'll focus more on results, which should be detailed, accurate, and chronological, as I have already mentioned. But the important thing about this is that you have to communicate your results. So this is the heart of your paper, always remember, and it needs to be presented, or else it cannot go into broader readership. And you can see there are nice article by Professor Kamatz on this particular series, and then you can go and check up this reference. So this needs to be presented, and then there are two major things that one should focus. One is accuracy in terms of scientific context. The second is aesthetically presentation. So basically what it means is that it should appeal to a larger audience so that your paper gets to a lot more people than we are generally used to. So, and the reason why this has happened, that there's an increasing growth in the number of papers being rejected on a daily basis, is because if you do not show your data properly, it's often equated with very sloppy scientific temper. And this is a fact, and you can go and check on, let's say, any big group. For example, Professor Whiteside's group has a particular segment dedicated to this, because with so much work being done these days, people do not really have the time. That the reviewers will just look at one of your figures, find a mistake, and then that person will not read your paper at all. So try to focus on your figure, because the first thing people look at is how people have presented their data in the form of figures or schemes or illustrations. So do that, and then you have already got the person to start reading your paper. So now I go to the importance of the figures, and then there is a cartoon, and here I've just tried to put in some old Tintin references, which I used to like as a child. So Professor Calculus is saying that here's some interesting data, and you'd like to see, hear, or read about it. And Captain Haddock replies that you'll remember if better if he sees, because seeing is believing. So when you present your data, the main thing is the content, the statistics that you show, and the design, how you do it. And then you present a lot of complex data, and that needs to be done with clarity, precision, and efficiency. For clarity, it means that there should not be any doubts when one presents the data. Once that is done, people should not have a lot of extra questions of, okay, this has been missing the figure, so what? So it should be very, very clear what you are trying to show. Precision is the truth about the data. So you precisely present what you have found, and not something more or something less. It has to be exactly what you have found. And efficiency is how you present. where you remove chart junk which has nothing to do with the figure. So these are the three ways, or and you can find it in this particular reference when you go. And there, I'd really suggest you to go and read the Tufte Edward visual display of quantitative information. A lot of the thing that I'm presenting is inspired from this particular reference, and also Professor Prashant Kamath's research tips and Professor Whiteside's research group slides. So I just wanted to mention that in the beginning as I go along. And then comes the aesthetic value, the way that you present your data. It looks impressive so that people are naturally inclined to read your paper to see what you have done. So like this paper has mentioned previously, it has to be the greatest number of ideas in the shortest time with the least ink in the smallest space. So basically, you can see they're all superlative terms, but it is very true. And you'll be surprised as I go along with the examples that I'll show you in a moment, how effective the communication is with figures and illustrations. So how do I present my data? The first things first, like the most effective or the most common one is the figures, and figures can be of a lot of types. We have drawings, we have diagrams, we have images. Drawings are basically realistic and accurate depiction of something. Like, for example, you have made a setup in your lab and you want to have a diagram. So basically, these are like three-dimensional blueprints of what you are doing. And then diagrams are very simplified. For example, you are drawing a circuit, or for example, you are drawing something which is very, very schematic of one elaborate process, which if you put in more details, which will clutter the figure. So you make it much more simple so that it communicates with you in a much more easy way. Images are direct visuals, and we see them all around us all the time. Graphs are what we are used to see more in our scientific journals, but I'll also go through some of the other ones. Maps are very efficient for spatial relationship, and they are very, very communicative because it's a visual thing, and we are much more used to seeing maps historically. And then you have flowcharts, which is also used as a schematic to show the flow of things in one particular direction. So suppose there is a sequence of things that you did. For example, you are doing a reaction which has 10 steps. It's very easy to show it with a flowchart. Then we have tables where you put in all your numbers. It has to be very, very precise. And the good thing with the tables is that you can put in a lot of different units. Then finally, I'd say a few words about the table of content, which is which has come up over the recent years. It was not there previously. I'm sorry about that. For the table of content, these are basically a visual storyline, so that an ideal TOC should reflect the title of the paper, so to speak, but we should not overemphasize it. So coming to the tables, so the idea is it has to be precise and one has to look for a sequence. So it's divided as a title. So first I'll go up. So on the basic format on why we use tables. So it's very effective in comparing individual values. For example, you're reading a paper and you want to have a reference to one particular event or one particular parameter. You can easily go back and check the rest of the parameters for a table. But it requires accurate values to be present. You cannot change things in the table. For example, you have fit a data or or you have a parameter that you are tabulating, it has an error limit. You cannot miss the error limit. It has to be exact. And also, the other important thing about the table is that you can tabulate a lot of different units. So basically, it gives an overview of different parameters at the same time, which is hard to do as a figure. For example, I can show you that for a table, always like there is a title and there is a caption. Very important is a footnote. If there are something that you have taken something from a reference, do not miss the footnote. That is very, very important. Then you have a column spanner. It actually expands the column, make it easier to read. You have column heads, and you can also provide dividers, which makes it easier to see. And then you can separate the table out in different segments so that it's easier to view. These are the stub heads and these are the stubs. So these are segments in the table body that you can look into separately if you want to look at one particular segment. For the other ones, what we would like to do is that try to get rid of the grids. When we think of a table, initially the format was that you have rows and columns separated by grid lines, but grid lines are visual disturbances. They create more clutter when you have a lot of things to tabulate. When you have just 3 rows and 2 columns, the grid lines work perfectly, but when you have 10 columns and 12 rows, grid lines actually cause visual disturbance because it takes up some space. So move towards free form, get rid of the lines as much as you can. You can try to use zebra stripes, which are basically alternate black and white stripes. You can use a header contrast. These are just technical things that can help reading a table a lot when you are trying to put in format. Always remember that your paper is a 2-column or 3-column format, and when you're trying to put in a table, the letters are very, very small and very, very confined into a small space. So you have to clear the clutter in all possible ways, and these small things come into effect very large scale when we are talking about this. So you have to put enough padding so there is enough space around each of the letters, and then do the tabulation in order. So maintain a sequence so that the reader finds it easy to communicate. So the hack here is minimize visual noise. I go to the figure. I have talked about drawings, and drawings are very realistic and accurate, but it requires a bit of artwork. like if you have a good program that creates good 3D images, it helps a lot. But it is used much more these days in the form of 3D viewing. But remember, it provides less scientific information. It's hard to follow a figure. This is more useful for technical writing, where you want to, for example, this is a schematic of an analyzer for photo emission, and then you can see exactly what the process is going on, but it does not give you the basic fundamental working principle of photo emission processes, so to speak. So, but it gives you all the technical information that has gone into to build the spectrometer. Diagrams, like I said, are very, very schematic and simplified visuals, but then people have evolved and now they have made diagrams which are really, really 3D, but they are much more informative. Then we come to images. We see in the scientific journals and in our daily lives, for example, medical imaging, we see it all the time, and it's basically scientific data that you are seeing. But here, the resolution is very, very important. Resolution can really kill your images. Then you have a flowchart. For the flowchart, the key thing is that you use consistent design elements, which means that try to use the same font, similar type of wording so that it's easier to follow, and try to keep the scheme on one page. And when you have that flowchart in one page, you try to write what's in the flowchart in the very same page, so the reader doesn't have to overturn the page and look back and forth all the time. The direction of the flow is always from left to right and top to bottom. And if there is a return process, try to do it from top to bottom, because that's how naturally we try to read a thing, from left to right and top to bottom. But the main point, how you want to communicate your data, is what message you want to provide. So if you define your message, you get to know why you want to show and how. Maps are very important for scaling down large information, spatial relation between parameters, and it communicates to us very, very easily, and it's very, very immediate. I'll just give you an example. For example, like the chai story, I'm sure you already know that the tea that we have is only known by tea and chai in different parts of the world. So basically, when you have the map, you know that it's because of the trade route. So wherever these leaves have reached by land, it's known as the cha or chai, and wherever it's by the sea, it's known as tea. So, for example, traffic updates, we use our Google Maps all the time. So how does Google give traffic updates? The maps, you can see that this is the red line. This is very, very highly jammed in traffic. And then there's the orange one, which is a bit less traffic, and the blue is where you can go free. So of course, you're close to the airport and there's a lot of traffic. So the way Google Maps does it, that it takes a historical data of the average time that is taken by a car going from one point to the other. And then, but how do they create the historical data in the first place? That is by the real-time data that is sent by sensors and smartphones. There are sensors in your laptop, camera, everything has a sensor, and it calculates how the position has moved as a function of time between the 2 points. And once the data is collected, it's immediately thrown into the historical data. So basically, this process goes on in a loop, and this map over a period of time gets much more accurate. And using this, Google Map can now it is also predict accidents that has happened on your driving way. One very recent examples, and we're all in the middle of a pandemic, which is the reason why we're having this session as a webinar format, is that it was maps that was used as contact tracing, like previous pandemics as well. This was Wuhan, where the coronavirus started, and it was contact traced back to a flea market, which is basically a seafood market, and this had a very high density like railway station, from which thousands of passengers passed out in different directions, and then that's how the virus got spread out. But it was done through contact tracing by collecting data of where the thing spread, and then you trace back the origin, and it could have been traced back to this market, depending on the density of people infected at different time scales. So these are some real life examples how like data is all around us all the time, and we are using it to decode different problems that we have and creating solutions for them. I come back to the image, which is like in the main frame of the talk, and then the first things first is that be very careful about bad experiment. The resolution is dependent on the number of pixels in an image. It's the smallest unit of digital information. So the smaller your pixel size, the higher is the resolution. But look at experimental artifacts. These are very, very important, that you identify a bad experiment. And also, like a lot of instruments in different in institutes could have different technical limitations. Please be aware of those technical limitations before you start the experiment. And if you've done the experiment, look at the image very carefully to look for artifacts, because these are real difficult images to publish. Sometimes you have a fantastic and a top-tier instrument, but it generates very bad outputs. For example, this had no scale bar, and this scale bar was manually added later. So then you can have unnecessary details as well, which like if you put it in this format, it's already not readable in a slide. You cannot read it in a paper. It's much more difficult. I'll provide you an example. This is one case study, and you can see that this image had a lot of problems which was fixed. First is a scale bar, then there was a border which was provided, and you can see that these lines, these are like small wordings, were actually closed by borders. And then this was high resolution instead. So now these days it's a typical way of showing a TM image that you provide a high resolution information in the inset and give the relevant information about the particle size and the interplanar spacings, and you can put that. Of course, this can be improved further. This is not very readable. You can use white or yellow to highlight, because this purple doesn't stand out here very well, or the pink. So these are the ways that you can improve on a good experiment, bad instrument generated output. And the idea is that you have to guide the reader in the right direction so that he can immediately get what he sees. And also, when you have when you measure these particular things, try to check if the image is reproducible. This is one of the quick tests that everybody should make a habit of, and just test that, okay, my experiment is correct. And then you interpret, because if you do not know if the experiment is correct, then of course your interpretation depends on what you see, and what you see is wrong, then the interpretation is going to be wrong. The next thing is the caption that I briefly mentioned about. So how do you write your caption? For example, this is a three-figure panel, and then what you have to try to do is that the first things first, get your x-axis and y-axis accurate. You cannot make this mistake that you label them in a wrong way. So then you mention that in your particular figure caption. So either you mention what you have measured versus what, or you measure the name of the experiment if that experiment has a name. Like, for example, you have cyclic voltammograms here. Then you have to provide the most important experimental, theoretical, or fitting details that you have used to make this figure. So, for example, here, the scan rate is 10 millivolts per second, and this has to be accurately mentioned in figure A. In figure C, you have, one has varied the scan rates. This is from one of our papers, actually, and you have to mention varying scan rates. Then the other important thing is the color code. For example, you have to see that this is carbon nanotubes with manganese oxide, and then this is coded in red, and here in the other figure it's also coded in red, and this is in black. The bare carbon nanotubes, this is, the symbols are also in black. So things like this, when you color code, it's easier to follow. And also it's very, very important that you identify which curve is what, and in the caption it should accurately reflect which curve is what. For example, it's mentioned in black and wine color, and then you should have the same symbol colors. So basically, caption is everything that you are showing in your figure, and it should encompass all the details that has gone into showing this graph. So the hack here is it should be short and crisp, but make sure that you provide all the information accurately. So move to the axis now. Axis fixes are basically all you need to know how to fix your axis. And first is that you cannot miss out on the axis. I've often seen particle diameters and figures like this, histograms specifically, that people don't really pay attention on making a point that they want to see the y-axis, and this is not correct because if you do not have axis, you have no idea about the scaling of what your data means. Like, we have no idea what this height means, so to speak, if you do not have a proper y-axis with proper scaling. The second is one definitely has to identify the title, what these axes mean. So for example, these, as you can see, are the absorbance and photocurrent data, but then as a function of what? This cannot be missed. And also one has to do provide a proper scale bar, which I have already mentioned. On the axis is not important. The scale bar gives you the magnitude of what you are looking at, or and then only you can compare between two different spectra. The other thing which really creates a difference is that you try to close the frame. These are bare frames. This is the same figure, and I have closed the frame. And now you can use this as a multiple panel figure as well. But it's much easier when you have maintain similar aspect ratios for different figures. This is particularly helpful in multi-panel figure, so that people do not mix up between two figures. Suppose this is, you have not closed the frame and there is another figure next to it where a lot of data points are very close to this region and very similar color. From a distance, it'll become very hard to identify what is what. So if you close the frame, you immediately create a window for the people to focus on. The other easy miss, which I see very often, or we do ourselves, is that one should avoid meaningless ticks. If you think about it, there is no meaning for these ticks. We are plotting nothing on the axis, on the top or on the right, but we are still putting ticks. What do these ticks mean? These ticks do not correspond to any particular unit or any particular experimental data. What we are plotting is absorbance versus wavelength, and it's all on the left axis versus the bottom axis. So the top axis and the right axis. We do not need ticks to show anything because there is nothing on those axes. And the other thing is that you mention, like, you try to use consistent fonts on both of your axis and the labels that you're putting on the axis. So basically, if you make this too different, it's going to be very hard to read, particularly in a multi-panel figure where you'd have these problems, which will be much more severe. So as you can see, these are small details, but this can create a lot of difference. So the idea of the axis is it should be immediately obvious to the reader what is being shown in a given figure. So remember, axes are the backbones of your graph. Without that, we cannot define what you're showing. So the other thing, the next point is that the scale. So basically, one needs to know about the scale before plotting. So there are different types of scales, as you'd already know. These are absolute scales where you actually give the original values you found from your experiment. There are arbitrary scales where you use qualitative values, and then you can always use a relative scale. So you already have an initial value, and then you show the other values from other similar experiments with respect to that value. And then you have normalized scale, where you show part or whole of a data depending on how your data looks or what you want to show in your data, which I'll come to in a moment. So basically, one needs to have an idea of what scale one needs to use in the first place. Typically, for example, I can give you one example from this reference, that these are Arkansas football's wins as a function of how they did over the years. This, as you can see, is an absolute scale because we have absolute numbers. But then what you see is that if you plot the figure like this, it looks very different from the plot. It's basically the same plot, but in different magnitudes. And you can see this is because there's a disproportionate to the data range, because here the figure ends at, the axis ends at 12.5, which is somewhere around here, and it immediately looks different. So if you are an Arkansas football manager and you see this graph, you would think that they're doing very consistent over 15 odd years. But then in reality, they have some bad years around 2005, 2008, 2012, and 13. And the hack here is that you save space. You, for example, we often look at a data when it plots in a strange scale bar that we just zoom out and see how it looks. Basically, you do the same thing when you plot. You save space and see that you use minimum, you leave out minimum space on the top and the bottom. So this is a classic example where the data that you show is scientifically accurate, but it can be very, very easily misinterpreted. So this is the reason why scales are important. So when you handle scales over a large range, this becomes a bit tricky, because now you have, by definition, a disproportionate amount of things to show, and then your scaling becomes very difficult. But there are very easy tricks for that. Axis breaks are the first thing you have. So this is one plot, and in the x-axis you see there are weeks and years, and between 12 weeks and 52 weeks, which is a year, there is no data point. So you break it and make it more concise, or else in the graph it'll look like cluster of points to your left, and then it's separated out by a huge gap, and then you have a cluster of points on the right. So basically you separate them out by putting in an axis break. Very important thing is that you have to remember this is not a trend. So do not ever try to go and fit an equation out there, because you have broken the line. It's basically a step out there if you see it and the slope is different. So it's very important to remember that this is not a trend. These are just tricks that you can represent your data in a much better way. And when you have multiple data, you can merge it with an axis break. For example, you have a disk current and a ring current, which are very, very different magnitudes, and you can put them in the same scale by just using a break and still compare between the two. Because if you see, this scale is actually way, way smaller. This scale for example, like the ring current is so small that when you see the disk current, if you plot them together, the blue curve, which is the ring current, will appear linear. But now you can compare ring current and disk current, because using an axis break, you have separated out the interval space between the top panel and the lower panel very, very differently. You can change the position of the axis scale and move it here, axis break, and then you can compare. But it depends. The position of the axis break depends on what you want to show and how you want to interpret your data. So there are some other examples. A very easy example is a log linear plot. Suppose you have numeric data or something which is very linear and something which is a very large magnitude. It doesn't necessarily have to be a logarithmic function, but it's a huge magnitude and is jumping in the order of 10 or 20. Then you can choose the basis of your log into that and then transform it into a log scale. The log scale doesn't need to be as a function or like as a basis function to be 10 or e. For example, the log scale here, I've taken this particular example because they have taken 2 as a log scale. So then you can also have a log-log plot where you have both functions, both axes you can actually present in the form of log-log plot. And I've taken this example because you can see that people have done linear fits. You can always do linear fits, but remember, this is a linear fit to a log-log plot. So unless that is exactly what you're trying to show, do not miss and report that this is a linear function. This is a log-log plot, and when you transfer it to a linear scale, it will not hold. So remember to fix this when you report. You can always combine break, axis break plus log linear, axis break plus log log. Typically, one should be sufficient. So that's the hack I follow. I try to use only one and choose carefully which one is more effective. Does a log log plot give me something more than axis break? So typically I go for log log only when I try to see that there is a function that I need to fit in a different scale, and then I come back and report. Or else axis breaks should be sufficient. But of course, like if you have a huge gap in your data set, you should always go for axis break, and you can still use a log-log plot or a log linear plot. So this ends the first section of my segment, and I will go to the quiz break and questions. So before I go there, I would quickly come back to the panel and okay. So how do we go ahead with the questions? First of all, how is the talk going? So this is a quick feedback.
Feedback is excellent. Of course, the platform, Dr. Mukherjee, is quite general. It is great that you presented a different aspect of it, the art of it, the presentation of it. I think students, participants can freely raise questions because the platform is more general, actually.
Yeah. This is this was this was intended to be a much more interactive session, so that I get a feedback if people are easily communicating to what I'm trying to say here.
Yes, sir, about the polling option, we can create a polling here only with words. But if we mix up with words and pictures, then we have to go to the browser again for submitting the answers. So you suggest us, what should we do?
So I would.
Sir, we can take the response in the chat box also, sir.
We can take the response in chat box. I think that
by showing the question and taking response in the chat box, it can be done, sir.
Yeah, I think that is
so
something.
Mr. Kumar, you are suggesting, yes, to share the pictures here in the screen and taking the responses in the chat box.
Exactly, sir. Explaining and getting the feedback at the chat box only. Without going to any other window, it will be better to in the same window.
So I'm trying to share my screen now.
Yes, sir.
But for some reasons, I don't find it.
It is showing Dr. Soham Mukherjee has started screen sharing. But then
So
again,
kindly reshare it, sir. Yeah, it is coming, sir. Going to the question, sir.
It's coming.
It's coming.
Yeah, we can see a blank screen if that is what you are seeing as well.
Basically, I'm not seeing anything. So
this is a white.
So you
blank
have chosen
screen.
for whiteboard, I think.
Ah, you have chosen whiteboard, right?
no, like I'm just seeing my desktop for some reason. I'll try to
Just
see.
reshare it once again, sir.
Yeah, I can see the Zoom. Like I can see all of you, but for
Yes, sir.
some
Kindly share the screen once again.
The
Share
share
that
screen
window that
option.
Click on
Yes.
the, Dr. Mukherjee, click on that you want.
Yes,
Yeah.
yes,
Yeah, it is
yes.
yeah,
Now we see your desktop.
Yes.
So that
Yes.
is the screen you are seeing as well. So it is being shared, I guess. Yes, it's working because you just moved
Yes, sir.
some box. I mean,
Oracle
Yeah,
virtual box has been moved. Yes, sir.
but for some reason I cannot see the presentation that I'm trying to show.
So you can open the presentation by reopening the folder or the PPT.
Yeah.
Yeah, it is coming.
Yes.
No, it's
Quiz, break, and questions. Yes.
Yeah. Okay, good. So we're back. So I move forward. Yeah.
I have a question, Doctor. Clarification to seek for
Sure.
Dr. Mukherjee. You use the term stub head, and you also use the term column and column head.
Yeah.
Could you clarify the difference? I mean, I couldn't really.
Stub is actually a subsection of the column.
Okay.
So when
okay.
Yeah.
Okay.
So basically, when you say a column, you mention the entire column, but you can break it into even smaller stubs. For example, you have sample 1, and then you have sample 1A, B, C. So I would call sample 1 ABC a stub, sample 2 ABC another stub, but sample 1 and 2 together form a column.
Okay.
Yeah.
All right. Thank you.
Yeah, you're welcome. So I go to the quiz break, and then I'll follow up this session with questions. Sorry. So the first question to the audience is, what is wrong with these figures? There are 2 figures, as you can see. And then these are from, of course, different papers. So I will give you 1.5 minutes to see what is wrong with these figures, and then I'll wait for the time, and then I come back with answers. Does it sound good?
Yes.
Participants can put the answer in the chat box.
Yes. The instruction for the participants, please find the answer and put it in the chat box.
Anil Chamuha, outside border. Answer response is coming soon.
Yeah.
For participants, please put your answer in the chat box.
Yeah.
For question 1, what is wrong with this figure?
Yeah, that's a very good observation. Next.
Okay, sir, responses are coming. There are several responses in the chat box already.
Yeah, 30 more seconds. Sorry, I'm on a timer here just to keep a track of the time.
Okay.
Soham, you have kept all of us wide awake. 175 of them after, some lunch of rice and curry and all that. So
yeah, that I think
credit goes to, I must say, it is quite engaging. Thank you.
Yeah, you're welcome. I've tried to do my best to keep it as interactive as possible. Okay, I think I come forward with the answers. So this question is pertinent to the figure caption I discussed. So the biggest problem is there is not a very clear definition of what goes in the inset. And of course, somebody in the audience has pointed out, and I want to thank him or her, that there are wrong things, like they have broken the access barrier and things have come out. But it's a problem with the inset. Remember, in the figure caption, it is very, very easy to miss out on the inset, and one has to accurately label what things are going in the inset. In the second figure, the problem is the symbols are wrong. Here, these are circular, and here, these are in diamond shape. And as I say, these are very, very simple things to miss. So one has to be careful what goes in with the caption, because your figure caption is actually the caption for your figure, right? It should say the figure in words, so to speak. So be very careful with the caption. So I go to question 2. Which is, how much ETOH do you think forms after 1 hour? So this graph is only up to 20 minutes, but if I can always fit this data and extrapolate and see how much ethyl alcohol is forming after an hour. So what do you think the audience is the amount of ETOH formed? So I give you 2 minutes again, then I wait.
Participants can put the answer in the chat box for the question 2.
So as you can see that there are 2 different samples here. One is a bare ETH and another one is a processed. You can provide me the answer for any one of them. So how much of the chemical do you think is forming after an hour? Okay, shall I go ahead with the answer?
Yes, sir.
Okay.
Thanks, sir.
Okay, good. So the answer is you cannot calculate how much ETOH is formed simply because there is no scale bar. And this is the reason why I discuss so much about the axis and the scale bar. Unless you quantify what you're plotting, you cannot know what the figure is exactly showing. For example, you can at best show that since this is a positive quantity, you can show that the red sample is formed more because this is a positive quantity, but then you cannot quantify which one forms by how much at a given period of time. Forget about extrapolating. You can of course fit this data and then get information until an hour or 2 hours, but then you cannot quantify it. That is the trick. that plays when you have wrong axis labels or you miss out on the axis scaling and units and stuff. So this is something that I just glossed over, but I still wanted to know. Arbitrary units are helpful to compare between dimensionless parameters, dimensionless versus parameters with dimension, true or false, and why. So your time starts now. I give you 2 minutes again, and we see it all the time that we plot something in terms of arbitrary units when we cannot quantify it properly. It's okay to use arbitrary units, but when do we use arbitrary units? For dimensionless parameters, when we try to compare them, or dimensionless versus parameters with dimension. Which one is true and why do you think that is true? So do I get some answers? Any answers from the audience? What do you think about arbitrary units?
So there are lots of responses coming. Help in scaling the quantities. Option A, more suitable case. Both are false. Because dimensionless means dummy.
Sir, is the chat box visible to you, sir?
The chat box is not really visible.
You can just click on the lower side of the panel and you can find out the question answer.
There is a screen called chat, sir.
Yeah, but
there is a tab button.
Yeah, I know that there is a chat box, and
if you click on that, the chat box will open.
Yeah, it's Q&A, but it's somehow full screen in this computer, and then okay. So now I see. Okay, so there is a lot of answers, and there are actually very good answers as well. So now that I can see the chat box, I feel much more comfortable now that, okay, now I'm getting a response.
Yeah, yes.
Thank you, Mr. Prem Kumar, for this. So I would go to the answer on this. The first things first, that a dimensionless parameters does not have units. So even the term arbitrary units is not really correct to use there. And you also cannot compare like both option A and B with arbitrary units. Arbitrary units you use only when you plot something that you do not quantify the unit for, but it's very different thing to compare between two dimensionless parameters and dimensionless versus parameters with dimension. And arbitrary unit, in my opinion, is very, very overused in the general scientific practice. But if you look up like the definition of arbitrary units, and then there are seminars around this, you'll realize that how much it has been overused. The bottom line is use arbitrary units only for parameters that you cannot quantify. For example, counts for a given measurement that you cannot really quantify. You can use arbitrary units, but when you do the same for another sample, it's very different. It's very difficult to compare it with the previous one because you do not know what the counts were dependent upon. So you do not have all the parameters or all the control on your hand. So both are false. So the next question is, how do you improve this figure? This is an easy one, and I gave you a case study on this. And by so you can, I'm sorry, there is a misspelling here. And by improving, I also mean that what extra information you can have from this particular figure. Like, how would you help the reader as well? Not just to improve it in terms of figure, but like the information coming out of this figure as well. Yeah, there is one response, including pixel. You know, the proper title and sufficient information about that. Improving resolution, very good. Yeah. Full length of the object is not visible. Okay. But this is an instrument generated image on one particular section that it is seeing under a microscope. Contrast, very good. Contrast. Make it in true color. Put an inset, pixel information, improve resolution, give a title. Fantastic. So basically, I think the audience have got it exactly correct. So one has to improve on the scale bar. It is hardly visible. One has to improve the contrast, but try to avoid additional color. And one can, of course, put the particle size, average particle size somewhere in the picture so that people do not even have to go through the details of the figure caption as well. The figure itself should be immediate. But the biggest fix here, if you do not even give those additional information, the biggest thing is that it has to have better resolution, and the scale bar needs to be bigger, because without the scale bar, you have no idea what length scale you're looking at. This can be anything. So that is the answer. Then answer 5. This is a very, very famous image. Can you identify what this image is? And there's a hint, which is Jon Snow, and I tell you, this is not a Game of Thrones reference. No, this is a much more famous Jon Snow. So anybody in the audience and the panel knows what this image is, and why this image is one of the like most effective images that has been roadmap, street. Yes, but what is the importance of this image? Before I think this of course, as you can see, that this is a very old image when 3D viewing and map drawing was just beginning to start. So these are mostly hand-drawn things. These black boxes that you see around here, do you see my cursor, everyone?
Yes, sir. It is
Okay,
visible, sir.
Okay, so these black dots actually around here, these black boxes, are actually number of case studies, like number of so this is basically a scientific study map on a number of persons, and this black box represent the number of persons in that area. I call this black box. So you can see the increased frequencies around this red dot. Okay, so no, this is not a satellite image because in those days there were no satellites to have such an image. So this is the cholera outbreak map of London, and Dr. John Snow solved this case. This gave him, so he's known as the founder of epidemiology, and we are in the middle of a pandemic now, right? And this was one of the smaller ones. This is the London outbreak, and this red location is actually the location of a hand pump, and these number of people were infected with cholera, and he was the first to identify that cholera was a waterborne disease, and he solved the problem by removing the hand pump, which is at the red dot. So this is one of the most famous images in the history of mankind that solved a pandemic at a much smaller scale. But it gave, but the bigger like solution was it gave birth to the definition of epidemiology, and he's known as the father of I forgot the name of the street. Yeah, it's here. It happened in Broad Street in London, and this was solved because the data was collected. So this was the first example of data processing using a map and for a medical case study. So that is the importance of this map. Thank you for this engaging session, and if the audience have any questions, they can ask. Or else I move on in the next session, if that is okay.
Yes, I think so.
Okay.
Let us move on to the next one because.
Yeah. Yeah. So the next session is about the units. I've already mentioned it briefly. You have to always provide units for the systems that you're plotting, and there's a reference from one of our works, and here I have just masked the units so that you do not know. And it has to be correct. You cannot give wrong units. This is the same example which I just questioned. You need to provide scale bar along with units, or else you lose the quantitative information. The other thing is about the unitless parameters, which I just asked, and the audience have already answered. So you have to be very careful about unitless parameters. Absorbance is a dimensionless parameters, and you cannot use counts or arbitrary units for this. For example, normalized absorbance is also like, you cannot give a percentage to normalize something. So basically, I'll come to that in a moment, but the take-home message is that when you have the wrong unit, it suggests that the scientific temper is bad and creates a very bad impression. So a reviewer who is looking at a paper would immediately reject your paper if the unit is wrong, if he or she finds out that you have put a wrong unit, or as bad as like you have missed a unit. So if your units can be confusing, I can understand with different forms of presentation of similar parameters. So you have to verify if you're unsure, just check it and then go to your figure. The next question is about normalizations, because we of course often present a lot of normalized data, and I recap quickly again on absolute scale, arbitrary, relative, and normalized scales. Use absolute scale ordinarily for original values. Arbitrary is qualitative, which I just mentioned. And then relative is with respect to what you already have as an initial starting point. And then you have normalized, when you can do selective comparison, or you can do for the whole spectrum, depending on what data you are looking at. So, like I mentioned, is very overused and misused. For example, take this, there's a set of intensity as a function of wavelength, and it just says normalized intensity, but the question is normalized to what? We have different peak heights at different things, because you can see when you just say normalize to your program, whichever program you're using, what it does, it just divides the maximum of the data and scales it between 0 and 1. So basically, if you're looking at this peak height, there is no point in normalizing different data at different peak heights, because you can see the maximum intensity for this curve is here, maximum intensity for the other curve is here, and for this one is here. So it will, so if you leave it to your program, it will mess it up. So always think what you are normalizing to, and the second thing is why you are normalizing. And the good thing about normalization is that you do not have to apply it to the entire graph. If you have set of spectra which are separate from each other, excuse me, you can always do piecewise normalization to compare your data. For example, this is a classic example from one of these references, that when you're looking at the relative scale for the intensity, then relative scales are much more important, because you're looking at the peak height and you can see how it decreases. But when you are looking at how the peak moves as a function of something, here is wavelength, it's always better to use a normalized scale because it amplifies the peak shifts. So then all depends on what you want to show. So if you want to send a message which is like, I want to show the reader how the peak shifts, then you define your normalization criteria and choose a normalized scale, or else use a relative scale. So these are different ways and to plot the same data, and it all depends on what you want to communicate. But whatever you do, I think is very, very different that you define your normalization criteria somewhere in the paper, or else people will not be able to follow. Some comments about the fit and trend lines. When you try to fit the data, I'm always in favor that you put it in symbols and show the fit in line, and try to give the difference whenever it is very essential. That is something. For multiple plots, when you handle multiple plots, you try to give the same color code so that it's very easy to follow. If you have a 2D plot, in the sense that you have not a 2D plot, but you have both axes which has errors, do not forget to to mention both. Sometimes it's very easy to forget that you have two axes and you miss out the error. And then the fourth, which is very important, is a causality check, which is that is these two parameters are dependent on each other. And so for example, there is no causality between the different kinds of elements which are mentioned here with their energy values and the migration barrier. So this plot essentially means nothing. This is a graph disguised as a figure. This is not a graph. This is much better when you plot it in a bar diagram, because the order of copper, silver, iron things do not matter. You can interchange and get a different graph. So the trend does not mean anything. So do not use trend lines. And also some basic things like if you have two data points, you cannot fit. It's always going to be a linear fit, even for a trend line. And the other thing is that when you are showing a fit, try to avoid trend lines because they disturb the fits. So either use a trend line when it's really necessary, or you have a causal effect, or you use the fit, but try to use the color code, and always check the fit errors. And errors are important because of a very simple thing. As you can see, I'll quickly come to this number 4, is that measurement is the best estimate and uncertainty. So basically, uncertainty tells 1/2 of the story, similar to the best estimate. I'll give you an example. For example, this is one of the plots where you can see two different parameters. If you do not show the errors, it will signify that this was fixed during your fitting procedure, which may not be the case. If you have large errors, it means that this is much less reliable. And when you have smaller errors, it looks more, it means that your fitting is more reliable. So it's very important that you do the quick check on what your errors are. The errors should be provided the scale bar. It's the same thing, the same plot and the same. So I've used the same plot to drive you home the same message, that the scale bars should be the same for the errors. The font should be legible. You should not use too thin lines because it should be readable. Always think that this graph will show up in a 2-column or 3-column format somewhere in a paper which is of the A4 size. So it should be readable at that length scales. If you get that information, it's much better that when you write a paper, you take a printout and see how the graph looks, go back, fix it, and then check it again in a 2-column format. So when you do that, remember that error analysis is very important. You want to know what your errors are: absolute, relative, systematic, random error. Or for example, like I've just taken out an example of significant figures because this is not the part of the topic that I'm discussing, but it's still relevant. For example, when you have significant figures, if you have a value 1.234 plus minus 0.4, this has no meaning. This last decimal has no meaning because your error is at the second digit. So the right format is to use 2 significant digits that you have after the decimal place. So you have to identify the type of error when you fit, and then, of course, always report. The next one is an easy one. It's much better when you have multiple data sets that you plot and identify which spectra is which. It's just easier for the reader. And I would suggest that you can see that there's not an order in this. You try to arrange it in a better order. These are from these references, as you can see. You can use a legend and then mark it alongside. The key is where you do not mark it and there is just only the legend box. The better is when you have fewer data sets. It's much more effective if you label it directly on the figure. Then there is a dead giveaway that everything is very, very readable. But of course, if you have 10 different spectra here and you try to label, it becomes too clumsy and it becomes very difficult to see. For those cases, a key would be much more effective. If you have much more, if you have somewhere in between that, then, or suppose if you have much more data set to show, let's say 20 together, then it's hard to put in label for all of them in terms of legend. So use a key. So basically, this identification and specification depends on how many data you are handling, so to speak. So the hacks here are label as much and keep order. That's all you need, because identification needs to be immediately obvious. Fonts, these are a bit more cosmetic things, but can be very effective. It should be large enough, as you can see that in this particular plot, it's hardly readable, and also the resolution is bad, which brings me back to a previous one. Then it should be consistent. You can see that these two font sizes are very, very different, and it's harder to read. Use serif font, and by that I mean that the fonts which have additional I don't know what these are called basically, but it adds a dimension to your visual. So when you use, for example, Helvetica is one of the font which is sans serif font, and when you do a lot of figuring with Helvetica, it looks very boring and it's hard to see. But when you use when you use serif fonts, it's much easier to see. You can actually add in more text just because it's visually more communicative. So I have fixed this figure with all different types of things. You can see, use both upper and lower cases. Do not bold everything. If you put everything in block letter, it's very difficult to read. So use both upper and lower cases, and also use selective bolding. Like if you want to bold one spectra over the other, or you want to highlight something in your figure, maybe the figure like the sample name or whichever one parameter out of the rest, use bolding or italic so it stands out among a bunch of others. So this example the audience has already solved, right? You have missing fonts and labels, and then you can fix them manually, and you please use contrasting colors, something like this, which is more readable, or you can use this as a transparent background, but put in some color which stands out. So the resolution, sorry, yeah. So now I come to the coloring things. So the idea to use colors in a figure is that it should enhance, not distract. So as you can see, that is almost impossible to read the word distract, but not enhance. So I'll give you a couple of examples. You can see that if you use something like this to plot some absorbance versus wavelength data, these colors are not very reproducible. These belong to the same tone, and then the hues are so similar that it's almost impossible to read what is what. Try to use light background. Dark backgrounds are difficult to read because it absorbs a lot of light. So you would ideally like to have the structures in dark and the background in light. This is a very easy trick. Keep an eye on the grayscale. So how do you know if the contrast is good enough or not? So what you try to do is that you choose colors and check in the grayscale which one stands out better, because a lot of journals, the readers would read in grayscale. So always keep that in mind. For people with color blindness and other deficiencies, is always much better and helpful that you label them properly so that they have a guide to the eye, which I have made, and also have contrasting symbol types. So the square versus the circle, which stands out. Do not use something which are very close in each other and they're hard to follow. When you have a lot of them, try to use just lines. Try not to go into symbols because it looks clumsy. So the color hack here is you check the grayscale, how things turn out. Some correlation plots now, if you have two different parameters, and then you want to see which are kind of related, and then you want to see, you can use two different scales. You can use multiple y-axis or multiple x-axis, but always label them. And then use arrows to point out which one is it, which ideally I would make this axis red so that you have a red graph, red arrow pointing to a red axis and pointing to a red label. So things like this that makes the graph look easier. So the other thing is that when you have same units but very, very large different magnitudes, then you can also use the XYY plot over a normal YY plot. But always remember that these scales are actually very different. So try to be aware of spurious correlations. Your graph can look exactly the same as the other one, but they're actually very different from each other. And one needs to be very careful when comparing them, because you can see the baseline start at different points, and then the intervals are different. So basically this scale and that scale are very, very far from each other. This is just to show the trend that this is upward. So you have to scale properly. So now I come to some other interesting examples like histograms and bar column graphs. So basically, it's a quantitative data. For the discrete, you use histograms. And the difference between histograms and bar is that there is a causal effect. Like you cannot change the order of the histogram, whereas in here you are seeing at categorical data. So basically, order is meaningless. So these are things that you see very much when you see something like, for example, the cricket scores that you see runs per over, you always see in the form of histograms. But remember one thing, the bin width is very, very crucial because if it is too small, then you basically are going to data points. And if it is too big, then you can smoothen out the small details. So basically, choosing the bin width is actually the key for a proper histogram. And also for the bar, but then the gaps. So try to have minimum amount of gaps in the figure. So here the bin width is not that crucial. So choice between column or a bar graph. So you have a horizontal and you have a vertical one. So just see if you have a natural progression or an order. If there is an order, then you can go for it. But typically a bar diagram is more effective when you have like a particular order, increasing to decreasing or decreasing to increasing. Whereas a column, it's not really the case. But remember that this is a sequential data, and then you cannot interchange. So the other quick things are that like how would you write the things? If the names are long, it's much easier to arrange them as bars. So this is easier to read. When you have too many of them, this will be very hard to read. So it's much more better aligned to the eyesight. A quick comment on the pie chart. This is very effective when we want to show the fraction as a part of the whole. For example, like this is one funny example which I like. Pizza is a real-time pie chart of how much pizza is actually left because you have eaten it and it's round in shape. But you have to use smart colors, and for smaller pieces of information, try to follow order. This is a very famous internet image and like a failure of a pie chart. If you have too many segments and not put in order, it's going to be a complete mess. And avoid angular view because it kind of amplifies the region, the amount of data present, amount of percentage which you're trying to show. So try to use a normal top view, and is very, very effective when you have to show something as a fraction of a whole thing, which is 100%. So this comes to the next quiz break and questions. So I would like to quickly go to the mistakes. So just guess the mistakes here for the audience, and I'll open the chat box and I'll give you a minute to check because these are easy.
Participants, please go through the quiz, that is question 1, and according to the question, please put your answer in the chat box. Sir is giving the instructions for the questions. Sir, the responses are coming, sir.
Yeah, they're actually the audience has already guessed the mistake, so I'll just go ahead. It's a different font size, and then different. It's too close, and the biggest thing is they have missed to identify that there is a fit which is in red. So basically, this also needs to be mentioned as a legend. So this is very important that you do not miss out. And the red line, the fit line, is hardly visible. So it's very important that you use the right color and the right thickness of the line. So the next one is this an interesting correlation that so the red curve is Japanese passenger cars sold in the US, and the black curve is suicides by crashing of motor vehicle. So the question is, detective, what's wrong with Japanese cars? Okay, so actually this is a very famous example of spurious correlations. There is no causality at all. These are completely independent data sets and have nothing to do with each other. This is a classic example, like I said, spurious correlations, and you can just go in the internet and see how like how funny spurious correlations and how dangerous spurious correlations can be. So beware of spurious correlations. And I've of course getting responses from the audience, which is no causality, which is absolutely correct. There is no causality. The third is how do you normalize this set of spectra? So as you can see, this is some absorbance versus wavelength plots as a function of temperature. So using min-max normalization, okay. Yeah, so basically the answer is, what do you want to show from this figure? First of all, one needs to correct the baseline, and then there are different background functions for different kinds of plot. For example, in the photo emission, there is Shirley background, and this. So be very careful what background you choose when you normalize. Just do not normalize your data, because your backgrounds could be different, very well-defined functions. So look up the background first. Once that is done, you want to see which peak you want to see. Are you interested in seeing how the peak moves as a function of temperature, or the intensity goes down? My rough guess here is that people are more interested in the intensity. So I'd fix the baseline and then normalize it to one particular value and not jump around over different values. I'll pick one value and try to normalize it. So I come to the next question, is that, so this is average transaction by payment type. Which one would you pick? A proper bar diagram or a pie chart? What do you think communicates the data more? Remember that there is no thumb rule to pick which one is better than the other, but then some data presented in some way communicates better. So which one do you think communicates better? Yeah, I'm getting mixed responses on this one. But yeah, this is actually, the answer lies in bar chart, because it's given in an order, and the order is easier to follow. If this was given in in a disordered fashion, then pie chart would have been much more effective. So the answer is, if there is an order, it's much more easier to follow a bar diagram. If there is disorder, then I think pie chart would have worked better. So then the other question, in cricket, how would you compare a batsman's scoring areas and run chase of a second team? So suppose a batsman has made a century, and how do you want to see the scoring areas? Which scoring areas that he or she is batting at? And the second question is the run chase of second team. How like, for example, you have a 50 over game, and the first team has made some runs at 300 over 50 overs, and how do you want to see the run chase of the second team? So the answer for the first one is, of course, the wagon wheel, which we are all aware of. You want to see the scoring areas as a wagon wheel, don't you? Because it's a circular field, and it immediately gives an impression that, okay, where are my scoring areas? Whereas for the run chase, it will be very hard to follow if it's in the form of a pie chart, because you want to see the numbers in sequence, and it's easier to follow because it's a cumulative function. It slowly grows. So the audience has got both of this right. And I come to the concluding segment now. These are some examples of higher dimension plots, and you can see that you can improve on the effectiveness by using solid thicker lines, For the contours, I would say that you use contrasting colors as much as you can, use more gradient. These are waterfall plots, and these are very important when you have good start and ending points which are at different places. And for example, partnership between two batsmen, you always see in the form of waterfall charts. So the idea is that you do not lose the impression of the higher dimension, you color code it proper, label it properly. Same holds for the 2D and 3D plots that we have. These are used for correlation between one dependent and two independent variables. The idea is that you keep the offset angle, like the viewing angle, right, so that every axis is highlighted. Do not use very, very slant angles or very, very top angles so that you lose the other dimension. But the most important fact here is that it's very visually impressive, but does it add enough value? Always ask this question. Any plot would look better if you add another dimension, but do not force it. Make it simple. Go for high dimension plots only when you really, really need it. So some easy misses on this one. This audience have already shown that these are wrong labels. As you can see in the legend and the caption, they don't match with each other. These are very easy things to miss. Suppose you have discussed your data, on optical conductivity, and you have marked the peaks. Very nice that you have marked, but the figure caption actually doesn't discuss any of this. But then it's of course discussed in the results and discussion section, but you cannot miss out these details. You have to explain them in the figure caption as well. So you try to avoid repetition. There are some diffraction plots. You can see that there is not really too much value addition. If you plot it in separate panels, these are hard to read. And suppose you labeled one particular reflection, you try to label it only once, clear the clutter. Some other very, very easy misses are formatting errors. When you format your paper, look at the font type, size, image. If things have misaligned, misrotated, and moved to a different place, particularly if you're using a spell check and autocorrect, this can be disastrous. It can, like often, the way it works is basically it identifies what word is already in its library, and if it doesn't see the word Raman, and it would change. And this is actually a true incident from one of my friends who was reviewing a paper. It autocorrected Raman to Roman data, and it was very embarrassing to correct the paper. So please try to look for these mistakes. Figure sequence after revision, this can be a mess that the reviewer has asked you to like to remove a figure or add a figure, and then you have forgotten to change the figure numbers. So these are very easy things to miss. So try to follow these numbers, and then the citation numbers and the figure captions. So the same thing is valid. And for the citations, I have previously also mentioned that it has to be proofread and checked in 2-column format. So please do this. This is a very general hack, that you check things in a 2-column format. The devil is in the details. So when you handle multiple data sets, you have two basic keys when you have offset and overlay. For abrupt changes, try to use overlay as much, so it amplifies the change. And for the offset, if you have roughly similar intensities or similar differences between the graphs that you want to show or traces, use a constant offset. You can choose to go for a variable offset for unnormalized data, but then make sure that you do not make a comparative statement that this one is bigger than that. So constant offset and variable offset, play around with the offsets when you have handling multiple data. For abrupt changes, use overlay. And use of scale bars, this is one effective example that this scale bar is not mentioned because like these are different plots, but then the scale bar has been added manually later. So this is just one example. And when you have, you can also try to use 3D impression. This is basically a 2D graph. You can see that there are only 2 axes, but there is smart use of an offset angle so that the viewing is much better, and they have color coded it properly. So it's immediately very effective. So you try to play around with the offset as much as you can, then go for the other options is what I follow in general. Insets, I have already shown an example. It's always good to use the same color coding, the green for the green and the red for the red. You can use the zoom in function to emphasize. So these are ways to include one data within the other instead of doing it a different figure and without cluttering it. So with the insets, you can also do a fitting of different traces, and then these 4 data points are collected from by fitting different like 4 different traces. The idea is that the inset shouldn't be too small, and it must be described in the figure caption. You can also, if this is too hard to do, for example, if you have a lot of data, this as an inset of that one would be very, very clumsy. So you separate them out and do a multiple panel format, but make sure you use consistent color coding. Look how they have labeled each of them and each of the traces, and both figure talk to each other. So this is one example of one multiple panel, and this is very effective for a combined discussion when you have different experiment series and all combining and giving one particular conclusion. So this is a schematic, this is from theory, this is from experiment, but make sure that the discussion stay in the same page. If you have a figure like this and then the discussion goes in the next page, it's hard to follow, and the reader has to go back and forth all the time to see what is what. But this is an effective way of handling multiple data sets. You put everything together, and this is very effective when different data converge towards one conclusion. So the idea is that do not make this too complicated. If you think that this is really making a lot of clutter, separate them out. Absolutely. So this is the figure hack is a minimal chart chunk. So table of contents, this is the last thing that I would discuss. So this has come up in recent times and has been very effective, particularly in terms of promotional purposes and additional visual impact. So it's basically what it says. You have a table where you arrange your findings, contents, that is your major findings, as a figure. So arrange your major findings as a figure is the literal translation of table of contents, if you think of it in terms of like as a figure, how it would translate. So, and how do you find that the TOC is effective? So the idea is, can you predict the article, what the article is about? It has to be minimal and should be legible with minimum amount of text. Then do not make the TOC stand out too much as well, because if the TOC is very, very impressive and the figures in your journal or in the paper that you read are flat, it gives an impression that somebody else did the TOC for you, which is a very bad impression. So it should be consistent and should be reflective of the quality of the graphics within your article. So, for example, these are some examples of quality TOC, and you can see that these are just visual central themes of whatever your work is about. I'll just give an example from this figure. It's very easy to spot out that this is CO2 reduction. There's a paper on CO2 reduction. There is an imidazole ring with modified ionic liquid. These are things that you can directly see from the TOC. There is a gold electrode working at lower potential, and this is the title of the paper. And you can see the important features of this paper are all included in the TOC. This is another example where this is marked. As you can see, the X varies, so this is a systematic work. There's a color coding, so there's a chromophore paper. So these are small structural changes, and the system is yttrium manganese indium oxide. And this is the title of the paper. So basically, from the TOC, you would roughly get what the paper is idea about. So the take-home message is it should be clear, attractive, but you should not overkill it. You should try to make it as less clumsy as possible. So this brings me to the concluding round. So I'll just start with this since I am I have no more time. So I just quickly go through this. Which fit function can explain the current? So it's up to the audience now.
Santa, sir?
Pardon?
KL, sir?
Yeah.
Yes, Mr. Kumar. Yeah.
We are going for quiz break questions now. Yes, sir?
Yeah.
But before that, how much time I have, like a minute, 2 minutes, or
maybe 5 minutes, sir.
Okay.
Because we have to conduct one short question answer session after that, confusion clearing session.
Okay. Yeah, sure.
Yeah.
So okay. Then the answer is that you cannot fit this function because there is nothing on the x-axis. It doesn't mean anything, and also there are different samples. So basically, you cannot fit this function. So this one is an easy one. Which time series is more consistent? Since I'm a bit short on time, I'll quickly go to the answers. But yeah, I'll give you, yeah, a few minutes maybe. But you can see that these are lead iodine bond lengths as a function of annealing time and made by two different methods. But which series does look more consistent? I think the answer, the trick here lies in the error limits. You can see that the uncertainties are rather large, and given the range of uncertainties, it's very difficult to say which one is more consistent. Like I said, the error bars say half of the story. So in principle, you can fit both of them with just one curve. They're basically the same. So how would you clear this clutter? I think this is a question which is sort of repeating. This is related to the offset. So that's how I perceive it. So what do you think we should do to clear this clutter? Okay, so the idea is that there are two different plots around here, and then the other ones end much quicker than that. It's much better to show the ranges properly and try to separate this out. This gives an impression that the other data are actually segmented out by the inset. So the inset should not touch these borders. They should be a proper inset. And I would try to take this one out separately or make this range a bit smaller and put the inset on the top so it's much clearer to view. That's how I would proceed to clear this clutter. So there's a scale y-axis that is very important, that it has to be scaled. And then from the TOC, can you guess the idea of this work? You can see, so I'll just tell what this work is about because of time constraints. You can see there is a platinum catalyst. This is a CO2. So basically, this is a CO2 reduction process on a platinum catalyst. And you can see what they've mentioned in the title, mechanistic insights of CO2 reduction on a platinum. These I don't know what this material is in interfaces from inoperant spectroscopy. So basically, This is one example of TOC. And the last one is that, okay, yeah. In cricket, how would you compare bowling accuracies of two papers and batting partnerships? Suppose you have two bowlers from different teams or same teams, how would you compare how accurate they are bowling about their line and length to different batsmen? So for the first one, you use a scatter plot. So basically, we have all seen the pitch map of a bowler, but if you think about it, it's a scatter plot. Someone, Ritesh has already said accurately that it's a scatter plot. So it's basically a 2D plot, which I showed some examples of. And batting partnerships, this is actually not a bar graph, but a waterfall chart, because it starts and then it moves in different because it's a bar which has both dimensions. So it's basically a waterfall plot, not really a bar chart. So finally, you ever wonder how people shape different cars? We see a lot of fancy cars around us these days, but how are these car shapes designed? And the hint is Bezier. So I'll tell you the story behind it. This was first, so Bezier was a mathematician who designed parametric curves. When you have two different points, like point A to point B and point B to point C, and you move along these lines, the locus of these 2 points, they form different shapes. And by defining the endpoints of each of these, you can get different parametric shapes. What this was first used to design Renault cars in 1960s, and all of today's cars that you see, all of the vector graphics that you do are heavily dependent on Bezier graphs. So basically, it's a parametric equation, and you can do a lot of shapes, and car companies use this routinely. In all the things that you see, you draw an arc in your computer, they all use these algorithms to describe these line shapes. So basically, we again go back to the parametric equations and graphs. So with this, I would quickly recap. Your data needs to be communicated accurately and aesthetically, because you have to get a large readership. When you define what you want to present, it becomes much more easy to visualize how you want to show it in forms of figures, tables, maps, graphs. Some key factors that I discussed was resolution is a key requirement for publication. Do not use bad images. Caption, it should accurately reflect what your data is. Axis and scale, put things in perspective. So do that carefully. Units has to be accurate. This is also a key requirement for publication. For normalization, you choose what you want to show and then define the criteria, exactly mention it in the paper. Fitting and errors are very important when you quantify, but do not ignore errors. They are very important. Label fonts, offset, these are all to guide your reader for better viewing. Use them effectively as you go along. Coloring is for enhancement, not distract. So basically use as minimum as possible, just enough to help the reader to visualize it immediately. There are different forms of graphs that I showed, histograms, bar. So then define your variable, and that will define your which one to pick. For the TOC, this is basically visual summary of the work, but you mention only the major findings here. And as a conclusion, I would say that we encounter real-time data in our real life all the time. So if you look around you, you'll always find there's some sort of data that you are encountering, and just think about it and how to formulate things. Finally, I'd like to thank Professor Shukendra Samajgar for inviting me to participate in this prestigious event. Thank you, MAKAUT, in association with IUST for this wonderful event, because scientific and technical writing, I think, is very, very essential, which is largely ignored. And I'm happy to be a part of a forum which encourages younger researchers in this direction. Thank you, the organizers, School of Natural and Applied and Social Sciences and the Language Laboratory, for organizing this event. And thank you, TQIP third phase, for funding. And with that, I would thank you. And here's a pie chart for you. By the way, this is non-causal. Thank you so much.
Thank you, Dr. Mukherjee. Thank you for the vividly graphic, inductive session that we have just witnessed. Now, before we move over to the question and answer sessions, I would like to request the chairman of the FDP, Professor Dr. Shukendra Samajgar, to say something in his own unmatchable way. Sir, please.
Well, the visual impact has left me speechless. Well, jokes apart, this has been a remarkable session. For a few reasons, I would mention three. One is it has, at least in one aspect of this FDP, it has lived up to the title. That is, from A to almost Z. The entire spectrum of visual representation has been covered. The central part in my mind was emphasis on accuracy, then aesthetics, so that we do not get distracted. The icing, if it exceeds the substance of the cake, that is never healthy. And the third point that I would like to mention are the glossary of terms, which I personally have benefited from, that different parts of graphs and visual aids, they have such specific name. And unless we nominate, so to say, we cannot talk about them. Not only talk about them, it is very difficult for us to think about them in an organized fashion. So, for example, legend, key, and label, they're distinct but subtle applications. These are food for thought, and this should be used. We should be guided by such sophisticated thinking. So I would end with a quote borrowed from Dr. Soham Mukherjee, which is, label as much and make order. Thank you.
Thank you so much. Thank you so much, Professor.
Thank you, sir.
Thank you for his nice words. Yes, please.
And now, Dr. Mukherjee, with your permission, we will just move on to take a few questions from our participants.
Sure.
Okay, Mr. Kumar. Mr.
Yes.
Kumar, am I audible?
Yes, sir.
Can you please unmute Abhimanyu Bandopadhyay? Here's a question.
Sure, sir. So we have allowed Abhimanyu Bandopadhyay.
Mr. Bandopadhyay?
Can I interject here, please? there is a question from Milind Thakare, I think, and that is the question in the mind of many of us. That is, what software have been used? So if you touch upon that, I think many of the
So
that is something, and then we could go to the specific question. That is pretty much in everyone's mind.
Sure. I mean, the slides that I made are completely made out of PowerPoint, but then I borrowed a lot of images, like the impressive images from the internet and some scientific journals as well, which are referenced below my talk. But typically, PowerPoint should be enough, and you have to believe me on that. The entire part of my talk has been made with PowerPoint. But PowerPoint can be a bit tricky with 3D visuals. So I would suggest Adobe Illustrator, for example, for impressive graphics, and of course for structures and stuff. One uses other things. You can have, for example, I use Diamond, but then there is Vesta. But these are programs that we all know about. People use ChemDraw. There is Mercury, and then there are other versions as well. But if you are talking about impressive 3D illustrations, I think Adobe Illustrator does a pretty decent job. If you want to do much higher than that, then I have to ask one of my colleagues out here, because he does instrument designs and he makes super impressive 3D, which I cannot make. But I think
for plotting and graphing data, what are the good ones?
For plotting, I think like we have all grown up doing Origin. You can use MATLAB as well, of course, and then you can use Igor. These are the things that you can use in a routine way, which I think should be sufficient. Yeah.
Thank you.
Okay, Mr. Bandopadhyay, you can ask your question to Shoham sir, please.
Yes, sir. Actually, this is a bit practical question. So actually, I'll just read out my question first.
Yeah.
In some cases, it becomes very difficult to make experimental methods exactly reproducible for scientific code that is based on programming languages that are no longer supported, even though we revise it to the latest versions. For example, Python 2 as a programming language lost support since January 1, 2020, and now Python 3 is the only officially supported version. So since actually this is based on one of my use cases. So actually the majority of my work was based on Python 2. So how to manage and communicate such scenarios in scientific writing if one is concluding one's research?
So no, this is actually just not a problem pertinent to Python only. It's a very basic problem that we start to work with some program and then it becomes invalid at one point. I think one way of I am always in favor of saying things the way it is and not sugarcoat anything. That you mentioned that this part of this of the work has been done using this, but depending on
even though if we revise it in Python 3, I revised it for Python 3, and it can be used applicable in computational biology. But since in some scenarios it becomes very difficult, so that's why I was asking actually.
Yeah. So basically, I do not know about the exact problem that you're mentioning, but I understand like the way the question comes to me is that there always upgradation of the version and you lose some features and then you get constrained. And I have faced this problem myself. And what I do is that I just declare in the paper that this has been done using this version. Now, if the work is unfinished, then you are in a soup, because then you cannot translate the work already done in that version to this version. But then I think for that, the only question that you write to the Python people that this is the problem that I have, and they will be able to help you. Like just because a program is not available anymore doesn't mean that it has been it has disappeared. I would say that you write to them specifically, explaining to them the problem that you are having with the scientific journal, and they will help you out with that.
Yes, I've communicated with the developer, the original developer of the software, and they have revised it to Python 3. But still, there are a few issues with that. So, can we communicate it in the paper? If we, is it allowed?
I think it's very allowed. I mean, when the reviewer has asked you back a question, you explain it to them that this is exactly what has happened. I mean, what I'm trying to say here is that the reviewer is also very aware of these problems, because he's getting text like this every few days, so he's very aware of the problem. And this is not the problem that you are facing specifically. I think this is a problem that many of us face, and we just write to the like when you in response to your review letter, you just write that this is a problem that I'm stuck with, and he should be able to guide you on that. Typically, when you hit a roadblock like this, the journal would cut you some slack, and then he or she would allow you to publish until what you have already achieved so far. I mean, a technical constant of a program cannot stop the science from going forward. Is the way that a reviewer would see this problem, is in my opinion. But there are senior panelists here. You can ask; they're much more experienced.
Yes. Thank you so much, sir.
You're welcome. Yeah, somebody has mentioned you can go for Anaconda 3.
Yes, sir, but we are using Anaconda 3 only in that software. It is based on Anaconda 3, and I've also revised it, used it using Docker for making it more easy to deploy the software.
Okay, this will become too technical now.
Right, Mr. Bandyopadhyay, what I would suggest is there are some comments, it seems, in the chat box. You can get in touch with them. They seem to be more knowledgeable about the issue than the rest of us here. Okay.
Yeah. Thank you, sir. Thank you, sir.
But from the reviewer's perspective is what I want to show you, that perspective, because that is what I'm kind of discussing here. Yeah.
Mr. Kumar.
Yes, sir.
We have a question from Ms. Ray, Rina Rani Ray. Can you please unmute her?
Yes, sir.
Yes, madam, please.
Hello.
Hello.
Thank you for this illuminating lecture. My question is, what are the significances of supplementary data and metadata? Actually, journals often are requesting for this metadata. So, is it mandatory always? Or?
I don't think metadata is mandatory. I mean, typically supplementary information should be sufficient, but I do not know why exactly they asked for metadata, but it depends on the kind of journal as well, I think. And I am only acquainted with a specific segment of research area. I mean, what kind of journal and why they ask for metadata? If you could enlighten.
Actually, some Elsevier journals on biotechnology, they're asking for giving the details of the experiments and everything in the meta as a metadata. So,
okay. So I do not know how exactly to define the word metadata, because I personally give everything in the supporting information, and by that I mean I also give the fitting files, the link of the references that I have used. Well, for example, I have referenced some diffraction data, or like I've chosen something for modeling something as an input file. I also give the references to those input files, and I give my own files as well. Like I attach the program files, because if some reviewer sends it to another one who is an expert in that fitting procedure or looking at those data, he or she can actually download those files and look what I have done myself. I do not know if metadata does more than that, because metadata is to help the data that you already have. But I do not understand really how much important it is. But if they are asking you to do it, you have to do it. This is also for sure.
Okay, thank you.
Yeah.
Thank you, Dr. Mukherjee. I think, Director Sir, that it's 5 and we are running off. Can we take some more questions, sir?
Yeah, rather briefly, please.
Okay, sir.
Just 2-3 minutes, 2-3 questions, maybe
Okay.
however many, about a minute and a half each. Okay, people have been waiting.
Okay.
So
yeah, it's 5.
Okay, sir. Mr. Kumar, please unmute Mr. Ritesh Hazra. He has got a question.
Yes, sir.
Mr. Hazra, please ask your question to sir.
Hello.
Yes.
Yes, you're audible.
Yes, am I audible?
Yes, you're audible, Mr. Hazra.
Yes, thank you for letting me interact. I was asking about the reference to the graphical representations. So is there any type of guidance articles or books to which I can refer to in deciding what kind of graphical representation or graphical of the data I have to use for my own experiment?
Sure.
That is my question.
Right. And I do not know how this event has been organized in terms of sharing the slides. You can always, if there is a provision, you can take my slides and I can share it. But then, of course, I have tuned it according to the content of the topic. But you can go to Professor Whiteside's group, research group, and Professor Prashant Kamath's group, and you will see that most of the figures and things and discussions I have actually directly followed from there. They have provided very detailed information on how to, and they have segmented it out very nicely, starting from research ethics, how to write a cover letter and things like this. And JPCL has a journal, 20 by 20, they say it, on how to improve scientific writing. So that is what I followed. And also there are ample videos and on YouTube, TED Talks about this. You can go. What I've tried to do is to make the format more generic. So I have gone out of what they have already done. So what you see is actually a mixed bag of all different things to make it more interactive. But these are the sources that I would suggest you to go. And the book by Tufte, which I kind of shared in the link, that is, I think, the father of all of this from where it started.
That's the Bible.
That's the Bible, yes.
Okay, sir. Thank you very much for the information.
Thank you, Dr. Mukherjee. Now I'll take the last question from Mr. Sarup Kumar Ghosh. Mr. Kumar, please unmute Mr. Ghosh.
Yes, sir.
Yes, Mr. Ghosh, please ask your question.
Good afternoon, everybody.
Good afternoon.
Thank you for letting me interact with you. Enjoy a great session. So my first question is a briefing of result in terms of quantitatively, I mean some matrices is mandatory for in case of last 2 lines of abstract or some sentences in introduction part.
So in the abstract, you mean the central findings of your work has to be reflected in your abstract. That's the abstract. But then abstract is kind of you've all about like heard about abstract art. Is like, you hide a silver ball in the Ganges and put your hand on it so people don't see the ball. So it's to generate curiosity. It should be flashy, should be accurate, and should inspire the readers to go forward. That's what an abstract is all about. So you have to mention a couple of lines, but in the introduction, what you can follow is to, when you end an introduction, you say briefly what you did in your work as well. So that section will be slightly more elaborate than what you did in your abstract. That is the trick that is generally being followed, which I have noticed.
Yeah, thank you, sir. And my second question, I mean, right now in some reputed journal asked for sharing the research data, and there is a mandatory, checkbox are there. So how it is significant? I mean, I'm working in basically two process: image processing, medical imaging, and some bioinformatics, gene expression data. So so how it is significant? I mean, some reputed journal, some Elsevier journal. I mean, it is in information fusion like or expert system with applications, very reputed journal. They are asking for the sharing the research data.
So if it is a reputed journal, I think so it's okay if they ask for research data, because there has been a growing tendency of data manipulation and data misinterpretation. So sometimes what happens is like that you, for example, like I tell you have measured 4 different samples, and what you show is basically a trend by fitting something which is specific to each different trace. And then you do not show the plots because it's not really important, because it's the fit that you want to show. That's it. So what you show is actually derived out of what you measured. It's not really an experimental data. Then it's very important for them to realize and go through them themselves, or to check that what you have done is accurate enough or not. Sometimes it's very easy to miss, and we do misinterpret. For example, I showed in a log-log plot, one did a linear fit, and you report those values and you do not show the log-log plot. The referee can be immediately very confused about how these numbers are derived. So it's okay for them to ask your data files. And I myself would say that when you're presenting data, you do not have to share those details, of course. But then when you do modeling and calculations, you have to give these details. The second thing is that some people, like there are some segments of research, they really need this data to create database, and that is where you help them with sharing the data. Because for example, right now, perovskite solar cells, I think everybody has heard about here. They are creating a research database which is very connected to the Uppsala University, and we are a part of it. So we share our data, and we basically at some time read 20 papers and then say that, okay, this is the data that they have shown. And if that is not clear enough, that person is asking the authors directly, can you share us the data, although the data has been published. So what I'm trying to drive at here is it's okay to share your data, to check its validity and its importance, and to create a database for further research.
Yes, thank you, sir. And last questions. I mean, some page restricted journal, they are asking for, I mean, some supplementary material. So how do I prepare supplementary material? Suppose my manuscript content 20 to 25 pages, but this journal asks for the only 10 pages. So rest 15 pages I have to make the supplementary material. So how do I prepare the supplementary material for the journal?
Yeah, I think if you have written a 25-page paper, do not go for a 4-page paper format at all. That is a more comprehensive paper that you have written, and choose the right journal which allows a bit more space. But to come to your question, 25 to 10, it's not impossible, but supplementary information is not like you have 10 page and 15 goes to supplementary. You have to rewrite the paper in a much more concise way, remove the findings and keep it for a subsequent paper where you go in more detail. For example, a lot of groups, it's a very general habit that they find something very, very significant, and then they immediately write a physical review letters in the format of a letter. That's how letters work. They have very concise format. So they do not show a lot of data there. But then they go for a second paper where they elaborate, and then that is a 20-page paper. So you can always try to do that. There is no hard and fast rule that you have to put every single data in one paper that you have you are trying to write. Just try to work out your priorities and then concise it to the best you can. But what I would say, if you have 20 graphs, there is no point trying to fit that in a 4-page format. You cannot fit that. So that would lose its impact. So it's actually counterintuitive. You target a high impact journal and try to put a lot in. It's not that. It's the content that matters. It's the finding, the importance of the finding that matters. So when you focus on that and put the rest in the supporting information. The supporting information is the support. The backbone figure should be there. So choose the backbone figures. So choose which 5 are important out of the 20 that you mentioned to have a consistent storyline.
Thank you, Dr. Mukherjee.
Thank you, sir.
Now I would like to request Mr. Shushanto Koyal, Joint Coordinator of the FDP, to deliver the vote of thanks. Mr. Koyal, if you'll please take over.
Thank you, Mr. Sengupta.
Achha, before Mr. Koyal,
Yes,
give me a minute. There are, Dr. Mukherjee mentioned about sharing his slides. We thank him for that. Actually, the way this FDP is being organized, and it is for the participants as well, we had indicated that we put together proceedings of sorts, where the slides, instead of papers, it would be collection of the presentation materials with supplementary materials here and there, some of the references. So that would be shared with all the registered participants. It would be mailed to you. So just be a little bit patient. You would get it the way you got. I think everybody got the abstract book, didn't they?
Yes, sir.
Yes, sir.
We
It
have
has been
sent it.
forwarded, sir.
So similarly, in about week or at the most 2, the proceedings would be ready and would be mailed to all the participants, registered participants. Thank you. Now, yes.
Thank you, sir. So here ends the day 3 session 2. Now, I don't know if I am the most capable person to say the vote of thanks to such a beautiful presenter like Dr. Soham Mukherjee, but as Mr. Sengupta has given me the opportunity, I would like to thank Dr. Soham Mukherjee for this nice informative presentation, where we have got all the technical details, the aesthetic suggestions, and the communication through verbal representations. That's what I got myself. So for that, Dr. Soham Mukherjee, a big thanks to you from all the organization, on behalf of all the organizers, and a big thanks to all the participants for keeping patience from 2:30 to now it's about 5:10 or more than 5:10. And I can see here more than 100 people are still there to waiting for this session to be concluded. So a big thanks to all the participants as well. Yeah, yes, sir. And now, last but not the least, my heartfelt thanks to Professor Dr. Shukhendra Samasdar sir for planning such a beautiful series of sessions, such a beautiful FDP for all of us, for me as well. So a big thanks to all. And with that note, I would like to conclude the session. Thank you all. Thank you,
And
sir.
thanks to Mr. Prem Kumar and his team as well.
Yes, sir.
Yes.
Thank you so much for supporting us.
Yes, and are we going to? Is there any whatever feedback or something link? Some people were asking.
We are about to announce it, sir.
Okay.
Yes, Mr. Kumar has already shared the link in the chat box.
Okay.
So we will be receiving the feedback. And
okay,
Mr. Kumar, have you put the joining link for tomorrow's session?
Yes, sir. Yes, sir. So the feedback link for today's session and the joining link for tomorrow's session. And also we request all participants, those who have not joined our WhatsApp group, to kindly join the WhatsApp group so that we can easily communicate with you, because we are facing some problem in the mails, that few people are getting the mails and few are not getting the mails, because there might be some mistake in the spelling of the Gmail to Gmail. So that's sort of problem are there. So please join our group.
It is always a good idea to have parallel communication pathways.
Yes.
Good. Well, thank you, Soham. On a personal note, it was really, really good, I must say, all of us. And it was very well balanced and visually pleasing. And you taking the time out of your busy schedule, we really appreciate.
the honor is actually mine to be a part of this wonderful event. Thank you.
And I must thank a little bit, although in absentia, Rudro, because he only suggested you.
Yeah. I'll probably call him in a day or two. But
yes, I think today is his joining day. So
okay.
Today is his joining day. So
I'll definitely call him. Yeah.
All right then. Hope to meet in person someday. Okay.
Absolutely. Yeah. Bye-bye.
Thank you, sir. Thank you.
Thank you, sir. Dr. Mukherjee. Thank you, sir.
Thank you so much. Yeah. Bye-bye.
Bye.
So Shantikar sir, shall we close the session or we wait for the link so that they can take out the note of the link?
You have shared the link, na? So
yes, sir.
We can wait for a minute to for the participants to take the note.
Yes, sir.
Yes, sir. We will close the session. And you please, Mr. Kumar, please send the link to the WhatsApp group as well. As you do the YouTube video link and the joining link and the feedback link, all the links that you do every day. Please do that. Yeah.
Thank you, Shushanto.
Thank you, Shashoto. So I'm taking my leave.
Okay, yes, I will do the same. Thank you, Mr. Kumar.
Thank you, sir. So, for all participants, please take note of the links and please, who have not joined the WhatsApp group, please join it so that we can communicate with you easily.